Hello, everyone. Sorry for the minor delay, a couple of technical issues. My name is David Hendrick with TSA, and we'd like to welcome you to week 2 of the 18th annual and first ever virtual Symposium on the Conservation and Biology of Tortoises and Freshwater Turtles. We'd like to thank everyone that joined us last week, and we want to thank our sponsors for supporting the symposium. And we hope you enjoy this week's sessions, which are going to be bigger concepts and reviews and status assessments. First up, from Hawaii, Robert Thompson. Take it away, Robert.
Okay. Okay. Hello, everyone. Good morning from Honolulu, and thank you for being here. Today, I'm going to be talking about some new work on global diversification dynamics in turtles that has really arisen out of this now multi-year effort to assemble a large phylogeny for all turtles that Phil Spinks, Brad Schaefer, and I have been working on. Now, before I get into the details, I want to start by saying thank you. Thank you to TSA specifically. Assembling the necessary samples for this tree has really depended on this group of people. It truly wouldn't have been possible without your help and many others, so we appreciate it. So this is what the tree looks like. It samples 80% of the extant species of turtles. We got all the genera in it represented, except for 2, and then all of the families. The data here are coming from 15 nuclear loci. And we're at the point now where the tree is sort of in hand. We have this nice statistical estimate of evolutionary history, including timing. And now we're at the point where we're starting to use this tree to address some evolutionary questions. The one I'd like to talk to you about today is a really simple one, and that is, why are there so few turtle species? Why are turtles so undiverse? So we all know that turtles are an ancient clade. They're much older than many other of the major lineages of vertebrates, and yet they contain a tiny fraction of the species diversity. So if we take the number of species in each clade and just divide by the age of that group, we get some sort of species per million years. And based on this, it sure looks like something different is going on with turtles. And we were interested in trying to dig into this and maybe understand what's happening. So we can dig into the literature, and most of the relevant work here really comes from the fossil record rather than from molecular work. So I'm showing you now some classic work from the early 90s from Hutchison. This is based on fossil turtle communities in the Western United States. And what I'm showing you here is simply a list of genera that are known from this formation. The black bars are time periods when fossils have been found. And we see, as time runs along, right around 34 million years here, we seem to have this extinction event in the fossil record where many species or genera disappear all at the same time. Now, the interpretation from this work is that this is really arising from the cooling and drying process that was happening globally at this time. So the transition from the Eocene to the Oligocene is known as a period of cooling and drying, and specifically in Western North America as well. Other work from the fossil record has taken different tacks looking at these associations between environment and turtle diversity. Here's an effort to model paleoclimate envelopes of fossil turtles. And what we find here is an association between warm mesic habitats and more turtle diversity. Okay, so on the one hand, we have cool and dry and decreases in diversity. On the other hand, we have warm and wet and more diversity. We start thinking maybe there's a role of climate in regulating the diversity of turtles. And we wanted to try to investigate that further using the new phylogenetic tree that we have assembled. Okay, so to explain how we do this, we really just need to start from a simple hypothesis. So let's imagine for a moment that regional extinction event in Western North America, rather than being regional, is actually global. So if that were true, we might expect to see a slowdown in diversification rate at the point in this tree that corresponds to the Eocene-Oligocene transition. Okay. So the model here is relatively simple. Essentially, we draw a line down the middle of the tree. We estimate rates to the left of that line and estimate rates to the right of that line. If diversification rates go down to the right of the line, we may view that as being concordant with this idea of an extinction event at the end of the Eocene. Now, that's a very specific hypothesis. We don't want to restrict our attention to that. For instance, maybe there is a global slowdown in the diversification rate, but we got the time wrong. Maybe it's a little earlier, right? So we want to be able to account for that hypothesis, or maybe there's more than one break in the tree, and only one of those is the one that we're sort of focused on right now. And in fact, if you think about it, there turns out to be an infinite number of these sorts of models that incorporate different numbers of shifts in the diversification rate, as well as their timing. Okay? So in the face of this model uncertainty, rather than focus on any one model, what we do is use Bayesian methods to integrate across all of them together and then average those results in proportion to their fit to the data. This is what those results look like. So I've dropped a red line into this tree that corresponds to the Eocene-Oligocene transition. And the chart in the bottom here sort of shows our average diversification rate through the history of this tree. And we have a long period of relatively constant rates, punctuated by one roughly threefold speed up that starts in the Eocene and seems to happen right around that transition. So that was sort of a head scratcher for us. We saw that and like, well, it's kind of the opposite of that pattern from Hutchison. So what's going on here? So to dig into this, I want to just sort of walk you through a few facts. So this is a map of global diversity in turtles. Warm colors are more diverse. And the first thing I want to point out is these studies from the fossil record, as we all know, come from a highly incomplete source of data. The fossil record is highly incomplete and biased. So specifically, this Hutchison paper comes from an area in Western North America that essentially doesn't support turtles today at all. The paleoclimate paper uses more global sampling of fossils, but again, we're very restricted in our attention to specific areas. So it's certainly possible that something different is going on with the extant diversity that's represented in our tree. The second thing I want to point out is this period of global cooling and drying towards the end of the Eocene was sort of a major event in the history of the Earth, in that it changed many other things, including sea levels. So here I'm showing you the changes in temperature across time as well as sea level. And you can see there's major decreases in sea levels, like on the more than 100 meters. So this is a much larger decrease than we tend to think about today with climate change. And these magnitude of sea level changes really have an impact. They reshape coastlines and really change how habitats work. So as an example, here's a paleo reconstruction of coastlines in North America. This dates to the KT boundary. And there's a few things I'd like you to notice. First, in the middle of the continent, we have the sort of remnants of the Western Interior Seaway that is now drying back. And along the Gulf Coast and Atlantic seaboard, we see a coast that's really quite different than what we see today. If we let a little time run by, we now see some change. So the Western Interior Seaway is essentially gone. There's a little bit of warm mesic habitat left in the Western US that's supporting that end Eocene community of turtles that are about to go extinct. And the Gulf Coast is starting to look more like the present day. For example, Florida's a thing, even if it's not connected to the contiguous continent. And then it's not really until 10 to 5 million years ago that these coastlines start shaping up in the form they are today. Okay? So that's sort of one thing. We have this big change in coastlines between the Eocene and now. The second fact I'd like to point out to you is that if you spend a lot of time staring at range maps of turtles, and I'm sure many of us do, you'll notice a lot of them seem to hug coastlines. So this is the range of the Florida softshell. It's essentially restricted to areas that were not emergent before the Eocene. They were underwater. Here's Pyxis from southern Madagascar, tightly hugging the coast, Badager from Southeast Asia, even whole clades. These are the map turtles, very near and dear to my heart. And these are essentially a series of river endemics across the Gulf of Mexico. So we see this pattern of changing coastlines and many turtle ranges being centered in areas that were marine habitats 35 million years ago. And that sort of raises this question: might these global changes in sea level help explain the modern diversity of turtles? Okay, so that's sort of a new hypothesis. And to test it, we did something relatively simple. So we took our tree and then we went through all species of turtles and classified their ranges as being coastal. That's the left column here in green. So these are ranges that are almost entirely on coastal plains in areas that would have been underwater. Inland in the right column, so these are things that would have been emergent at that time, or widespread, so they're both coastal and inland together. So once with this data and our tree in hand, we can now fit a new type of diversification rate model. And this is just sort of a graphical picture of how this model works. So what we're doing is allowing turtles that live in different areas, coastal versus inland, to have different rates of diversification. Okay, so if you look at the green circle here, in coastal areas, we can speciate at some rate, S0, and that adds species to the coastal pool. These species can also go extinct at some rate, X0, that takes species away. The same for inland, and if you're widespread, you can also speciate and go extinct. Finally, you can disperse between these ranges, so ranges can evolve across the course of the tree. Okay? Now, this is the simplest version of the model. I'm not going to get into the statistical details of this. It's just too much for a short talk. But again, there's many possible models here, and we explored a large set of them and averaged across that uncertainty in proportion to how well they fit the data. So this is what those results look like. In the left box here, I'm showing you the net diversification rate for the 3 different areas. And sure enough, we see roughly 3-fold faster rates of speciation on coastal margins than we'd see in inland areas, whereas widespread is sort of intermediate. This change really seems to be driven by a change in the speciation rate. That's the right box. It seems that the overall diversification rate is higher because species are speciating more quickly in those areas. We can also use these models to reconstruct the history of shifts between these different habitats. So that's what I'm showing you here in the tree. The green tips are the coastal species. And one thing that really strikes me about this is essentially all of these transitions into coastal habitats post-date the Eocene-Oligocene transition. And that's demarked in the dotted line here. Now, the model has no real knowledge about timing of these events. There's no necessary reason it should reconstruct it this way. So the fact that it does it is suggestive to us. It's concordant with the hypothesis again. So taken together, we'd say based on these analyses, it does appear to be that modern turtle diversity seems like it may have been driven by rapid diversification on newly exposed continental margins. And it's really decreasing sea levels that explain both these patterns between the fossil record, where we have disappearances, as well as bursts of diversification in the extant species that we see overall in the tree. Okay, so I will leave it there with a few more acknowledgments to lab assistants over the years. Again, many museums and collections and individual contributors, many members of TSA. Thank you. As well as computation and funders. And if I have some time, I will take questions. Okay, thank you.
Okay, just a second. I've got to find my actual thing here. Okay. Am I on?
You are on. Go ahead and share screen.
Oh, well, okay. Am I sharing the screen now?
Yes.
Okay. What? You see the PowerPoint, right?
I don't yet. You do need to go ahead and share screen.
Oh, okay. Get back down there. I thought I hit that. There we go. Share screen. This one. Share. There we go. Now you should see me. Okay. And okay, here we go. You got the PowerPoint up now, right?
Yes, and you can go ahead and
Okay, I'm Malcolm McCallum at Langston University in the Department of Agriculture and Natural Resources, and I'm presenting on some research I've been working on over the last year or so. So this is about extinction of turtles and where we're at today. And so first, because there may be some people watching this that aren't scientists, extinction is, of course, permanent loss of species. Before 1813, extinction wasn't a given. It was thought of as kind of a possibility. People were debating whether things could really go extinct. And then Cuvier published his essay on the Earth, and that, of course, since then we've accepted that not only is extinction possible, it happens, and that the fossil record is that the fossils aren't just curiosities, as he put it, but they are, click the chat. Oh, my chat button didn't come up. Try that again. Chat. There we go. I do not see my chat button, by the way. The chat isn't showing up here for me. Anyway, and it's an important ecological and evolutionary process, and it's often confused with extirpation. Extinction is, it's gone. Extirpation is just a regional loss. So every species lasts about 4 million years on average based on fossil record. We know that most species that have existed have gone extinct. We've had about 4 billion of them, and that it's around that only about 1% are still extant. We can categorize extinctions based on 1 million, 10 million, and 100 million year events. These are minor, major, and mass extinctions. Of course, we've only had 5 or 6 mass extinctions in the history. There is some debate about a very, very early one. And these extinctions have involved a lot of very broad groups of organisms, or whether you're talking about placoderms or ostracoderms or dinosaurs or what have you. Each of these mass extinctions, the Ordovician, Devonian, Permian, Triassic, and the Cretaceous, were all 1 in 100 million year events. These were not things that happened very frequently. And they often spanned a lot more time than we think of. From a human perspective, these are huge amounts of time. Geologically, they're relatively small. Of course, the Permian Triassic, it could have lasted as much as 9 million years, depending on how you define what the Permian Triassic extinction is. Some people divide it into a major and a mass extinction. Some people combine them together, and they go back and forth on this throughout time. Of course, the Jurassic Triassic probably actually lasted about 500,000 years. While a lot of species went extinct initially, it still had species disappearing for quite a while, up to 2.75 million years, depending on whose estimate you follow. So background extinction, though, tends to be more background extinction tends to be more selective than mass extinction. That's extinctions we see all the time. Oftentimes, background extinction is balanced out by your typical background origination, which is the evolution of species. Background extinction, you see specialists go extinct, especially highest, super specialists that have real fine details in their life history, where generalist species tend to keep on going because they're very much more flexible. Over time, this just is to show you how background extinction on the yellow line relates to variation in overall extinction. So, I'm just trying to see something here. I do not have a chat box up. I cannot get it to come up. Our mass extinctions tend to be caused by abiotic factors up until recently, and that's because we are really causing it, even though we are causing geologic factors to occur. They also tend to be not very selective. The most recent mass extinction, of course, was the Cretaceous Paleogene extinction. Previously, we used to call that the Cretaceous Tertiary, but that isn't actually accurate because you're comparing you're using border of a smaller time period with a large, and you want to use the same time period category. About 76% of species actually went extinct. Of course, the dinosaurs, even though a few dinosaurs may have lasted into the Paleocene, that's under debate, but it's reasonable to think that could be. But unlike these previous extinctions, we basically have one species causing most of what's going on today. When we look at turtles, as of using the IUCN data on the Species Survival Commission, we see that we have approximately 8 species that went extinct since 1500. And these numbers may be a little off, depending on whose estimates you use. Extinct in the wild, critically extinct, endangered, in the different categories. Right onto data deficient. And of course, different levels of extinction, whether it's extinct in the wild, and so forth. It's important to realize that on fossil standards compared to what we see all the animals that exist or all the plants that exist. When you go into the fossil record, when ranges contract and abundance declines, the probability of extinction remains the same, and so you have them disappear from the fossil record before they disappear from the planet. Organisms that we count maybe 10 or 15, 20, 100, 1,000 species, individuals left, those aren't in the fossil record. They're already extinct. And they're classically called dead clades walking because they'll appear in the fossil record in a punctuated fashion. Just occasionally here comes a fossil, but there's no evidence of it being there before. Because we have this variation we have a lot of uncertainty in our data due to the fossil record, and also we have uncertainty in modern data because of issues with being able to revisit places. We actually have much more, I won't even go there. Because of this, when you're dealing with a lot of uncertainty, there's different ways to deal with it. And one of these that's very appropriate for paleontological data when comparing it with current data is using fuzzy mathematics. And what fuzzy mathematics is basically is this. If you think about a point estimate, which most of us use every day, we try to get a very exact number of how many people are in the room or whatever that is. And, a lot of times point estimates aren't all that important. Whether it is 33.235 whatever milliliters of fluid in his drink, or if it's 34, is it really irrelevant? Does he need to have his drink topped off? We don't know, because I don't know what the volume of his cup is. But we take a system like this, we're a wee bit low on the beverage, we right away know that we need to get some more. This is how fuzzy math works. It's giving you the trajectory. It's telling you that, hey, this is somewhere over here. It doesn't mean that an exact number in there is somewhere in that group of numbers is the right number. The bottom line is the stuff outside of that set is not accurate. If it's if you're trying to say there's no difference, and the fuzzy set showing enormous differences, saying there's no difference is complete bunk. It's not going to happen. We've all seen fuzzy estimates when we watch the hurricanes come into the coast. That cone that they show coming in, and over time they narrow it down. That is, in fact, a fuzzy estimate. So looking at the two, we can see that there are times when having the exact number is really important. But there are times when it's important to understand what's going on, and whether it's 2 or 3 doesn't matter. So from a mathematical perspective, it's set up like this in a distribution. We have things that certainly aren't possible. They get a value of 0. Things that are possible get a value of 1. And then we have things that are somewhere in between. And so on a graphical presentation, our fuzzy numbers tend to be triangular or parabolic in the system that we're using. They don't have to be. They can be wavy lines and all kinds of stuff, but that's more for people who are doing computer science and not for practical stuff like we're doing in this study. So then you get a set that has the borders, A and B, and then of course the middle set is the stuff that's most where we expect it probably is. And as you go from the M's to the A and the B, it's gradually becoming less of, those are things we can say are probably not true, but they could be. They're borderline. And we get a fuzzy interval like this. Fuzzy arithmetic's ideal for this, for looking at things where there's a lot of uncertainty, extinction estimates for sure. And that's about all I'm going to say about that. So how do our extinctions today compare to what we've seen in the fossil record? Well, there's a lot. I broke this up to modern extinctions since 1500 and since 1980, because we have some different things happening there as technologies improve. And so we compare it in that way. That's the standard way that people who've been doing this kind of research have been doing it. So we have all these extinction estimates based on places all around the world, a lot of them in the US, but also in China and other places. And then there's a series of standard equations that are found in conservation biology books and that for doing this kind of thing. Okay, so when we start, oh my goodness, I think am I on the right one? Wait a second. You know what I did? No, that's right. Okay. I was just double checking that I was in the right place. So as we go here, we can see that different species, different groups have gone extinct as of since 1500 at different rates. Okay. Now, but if we look at our turtles have gone extinct right here. This is what we expect, somewhere between 87 and 658 times what they did, what reptiles went extinct at Cretaceous Paleogene. And that's telling you, now that tells you a lot about what's going on. Again, since 1980, they've gone extinct much faster. Even now, this is considering that I'll go on and I'll continues. So remember that when we look at the IUCN data for turtles, it's a little bit different than what the TSA has posted. But the bottom line is that when we look at these numbers, they actually are comparable. And that's what we really want to look at. If we start adding in organisms that are dead clades walking, that are currently extinct, but they're going extinct, or we may be able to revive them, but they certainly are in very small areas and they're very endangered. We find that these numbers go grossly out of insane. This is the same kind of information, same kind of numbers we're seeing for other vertebrates, the same kind of numbers that Barnowski saw with Ehrlich and Sabalos, and the same kind of numbers that Pym has demonstrated. And in fact, when Barnowski put out his data using interval assessment, he actually came back and recalculated it, making assumptions more narrow. Just because people were complaining because the numbers were so high. As we look at this, so what would this mean for the sustainability of these turtles? How long would they last into the future? And of course, at the 1500 rate, we're talking about 1,000, less than 1,000 years. They certainly aren't going to last forever. They're not going to go extinct tomorrow. But when we look at the post-1980 extinction rate, we're talking about you could have them all go extinct that fast in 59 years. Now, the key here is that these numbers, the reality when you actually look at it, some species that are just like, red-eared sliders, they're going to hang around a lot. But a lot of your specialist turtles could disappear relatively quickly, especially as climate change ensues and so forth. And that we know that climate change, increased temperatures, messes with their physiology like mad. So a live animal may be sitting here and reproductively the population can be dead. So the conclusion, of course, turtles support the previous papers that have been done. And of course, that's basically where I'm at. So let me see if I can get this. Okay.
Malcolm, I don't believe we have any questions.
Okay.
So we can
go
on into Sean's talk next. Thank you.
How was my time?
You were pretty right on.
Cool. Okay. You have a very nice day. Thank you for having me.
Are we good to go?
We are, Sean. Go ahead.
Okay, thanks, Dave. Hi, everyone. I'm Sean Duty from University of South Florida, and I'm going to talk about synchronous hatching and emergence in turtles. And I'll especially refer to some adaptive hypotheses for why it may evolve in turtles. And if I can get this to page down, we'll keep going. Fun fact. So did you know most of what we know about hatching and emergence in reptiles is from turtles? So, and this is because turtles, some of them are gracious enough to leave a track in the sand like this spiny soft shell mother. Of course, some turtles are selfish and nest in vegetation, making it harder to find the nest. But the bulk of what we know about hatching and emergence in reptiles is from turtles. We don't know much about lizards in this respect. When was the last time you went out and looked for snake eggs? Never, right? Crocodilians, they're conspicuous nesters, but they always lay in places that are remote and hard to get to. You got to take airboats and stuff to get there. Then you have to deal with the mother, right, defending the nest. So we owe turtles for what we know about hatching and emergence. So what is synchronous hatching and emergence? This is going to evolve the definition as we go through the talk. Hatching and emergence are separate behaviors, right? But they can be linked. They tend to be linked. So you could emerge synchronously after hatching asynchronously and vice versa in all the different combinations. But traditionally, collectively, we talk about them as a simultaneous escape of offspring from the nest or nest sites. This can occur within or among nests, but I'm going to focus mainly on within nest hatching and emergence today, and you'll see why. And traditionally, we've considered these behaviors important anti-predator behaviors that are related to fitness. For example, the classic example is the mass emergence, hatching emergence synchronously of sea turtles, right? So each one of those sea turtles has its predation risk diluted, safety in numbers because it's surrounded by so many others in the face of predators such as crabs, raccoons, cats, dogs, armadillos, foxes, birds. And then of course, as all these turtles get to the ocean, they face a gauntlet of another suite of predators, fish, dolphins, etc. And there is evidence that this predator dilution, this predator swamping actually occurs, experimental evidence for it. But what about single nests of turtles other than sea turtles? So the picture you're seeing is all about among nests, synchronous hatching and emergence in sea turtles. What about a single nest of a non-sea turtle? Is there synchronous hatching and emergence? I'm going to use Pig-nosed turtle story as an example because it's what got me into this subject area. It's extremely interesting and unique. And I think we would expect, based on this story, to see synchronous hatching and emergence in this species. So pig-nosed turtles in Northern Australia lay eggs during the dry season. The eggs complete development in the dry season, but they do not hatch in the dry season. They go into this suspended animation where they're waiting in the egg for the onset of the next wet season. And so when we get heavy rains and rising river levels over the nests, the eggs then hatch. They've had to evolve explosive hatching, and you can see this on Life in Cold Blood from BBC, whereby pig-nosed turtles can hatch in minutes instead of hours and days, which we see in most turtles. So we've known for a long time that hypoxia is a cue for hatching in pig-nosed turtles. So the eggs go underwater, they become hypoxic, the embryos start moving around and hatch within minutes. In the late 90s, during my PhD, we discovered when we were digging up late-term eggs, that they were hatching in our hands. They were hatching in our hands, in the boat, in the car. It was a mess. And so we had discovered inadvertently a vibration cue for hatching. And we wondered why that would be the case. And as I held an egg and could feel an embryo moving inside it, I thought maybe this movement can expedite hatching in pig-nosed turtles. And so we did some experiments, and indeed we were able to show that groups of pig-nosed turtle eggs hatch and emerge faster in experiments than solitary eggs. And if you plug those data into a model using different river level rises, rates of rises, you'll find the bottom right graph that turtles that are exposed to a vibration cue will always hatch sooner than those with only a hypoxia cue. So basically what we're saying is that water level starts to cover the bottom of the nest, causing hypoxia, and those embryos moving around instantaneously send a vibration cue through the nest so that they can all hatch faster and get out in a flooding situation. So why did I drag you through an example that was published 7 years ago? Well, I think it's fair to say that we would expect synchronous hatching and emergence in this species given that scenario. And yet when we put cameras out on the nests, that's not what we found. So here are a couple of nests being monitored by the old Trailmaster camera systems. And you can see one pig-nosed turtle hatchling emerging in each picture. On the left, after heavy rains; on the right, after flooding of the nest. In fact, we lost those boxes from that flood, but of course the camera is separate here, so we were good. The interval between emerging hatchlings for pignose was 12 minutes. So we didn't really see synchronous hatching and emergence or synchronous emergence. Some hatchlings did follow 1 minute after another. Some took days to get out. So that was a bit surprising, and that, I think, kind of sets the theme for the rest of this talk, which is going to be reviewing the evidence for synchronous emergence in turtles in nature, and then using that evidence to scrutinize the adaptive hypotheses for why synchronous hatching and emergence evolves or is maintained. So what are those hypotheses? The first one we already talked about, predator dilution. Goes by a bunch of different names, gets very technical: attack abatement, predator swamping, predator satiation. Although it has a bunch of different mechanisms, it's essentially safety in numbers. A second hypothesis, called social facilitation, came out in the 60s to help explain how sea turtles, hatchlings, get together and in a collective fashion to excavate themselves and escape the nest. And some work out of David Booth's lab built on that recently and showed that there's a huge savings in energy in terms of the residual yolk in hatchlings due to that facilitation. So that's a really important hypothesis, at least for nests that are deep with lots of eggs like sea turtles. The next hypothesis is so little visited that it didn't have a name. So I've given it the name predator preclusion. And here we're talking about the fact that if you have, let's say, a painted turtle nest and your first egg hatches, it's going to give off hatching smells or odors that could attract olfactory-driven predators to the nest. And so it'd be best if everybody hatched and got out as soon as possible, i.e. synchronous hatching and emergence, so as not to attract predators through an asynchronous hatching scenario. And I'll return to that later. Finally, some of you might have already thought, do we have to have an adaptive hypothesis here? If we've got a turtle laying eggs that are experiencing similar temperatures, similar incubation periods, hatching about the same time, why do we need some adaptive explanation? And in fact, that is a good point for small turtles with small eggs and small clutches. In those turtles, we have very little layering of eggs. And so they will experience the same temperatures, developmental rates, incubation periods, and we would expect them to hatch and emerge at about the same time. And so things like box turtles, mud turtles, Sabine map turtles, all the small turtles that lay very few eggs or small eggs, we might expect synchronous hatching and emergence without any adaptive explanation. And if that's all we had with small turtles, then the talk would be over. But of course, we have lots of larger turtles with larger numbers of larger eggs. And so when we get these species, the three-dimensional space occupied by the nest is large enough to experience thermal gradients. That is, top eggs are going to be warmer than the bottom eggs. So mean temperatures are going to change moderately, maybe just a bit, but the fluctuations, the daily fluctuations in temperatures are going to change quite dramatically between the top and the bottom of the nest. And so we're going to get top eggs potentially hatching sooner than bottom eggs. And yet in the lab, especially, researchers have discovered that there is still somewhat synchronous hatching. So we've got these thermal gradients. We should have these big differences in incubation period and timing of hatching. And yet a bunch of studies have shown that in fact, that difference is not very large. So Spencer et al. Were the first to discover this with Macquarie River turtles. If you take eggs and incubate them at different temperatures and get some at going faster at a hotter temperature, and then you join the eggs back together, those least advanced siblings will catch up a bit with the warmer eggs that are ahead of them, up to 2 to 5 days earlier than controls. They still lag behind the others by 4 to 6 days, so it's not perfect, but there is some catch up in some species. And then a more recent study showed, instead of catch up, that some painted turtles can hatch at a less advanced developmental stage than other eggs. So there are different mechanisms here for trying, it seems, to synchronize hatching and potentially emergence. It's not really where I want to go with this review, and there's another interesting layer about how they might do this. They can possibly detect one another's heartbeats. Of course, we now know there are vocalizations, at least in Podocnemus expansa embryos, so there's a lot of neat stuff there. I just wanted you to understand the sort of framework behind why we might be extra interested in synchronized hatching and emergence. So what I did was looked at the data for 24 studies on 16 species of turtles. This is emergence data from within nests. The quality of the data are highly variable. Sometimes it's someone checking a nest once a day or once every other day. Sometimes we have cameras getting timestamps for every time a hatchling emerges. I ended up with 3 types of data or 3 sort of parameters. The percentage of nests in which all of the siblings emerged within 24 hours. The second one is just the range of that. So over how many hours or days did all of the siblings hatch and emerge from a nest? And then finally, for a couple of studies, with cameras, we had mean sibling emergence interval data. So let's look and see what we got. For the mean proportion of nests in which siblings emerged, all siblings emerged in 24 hours, we got 47%, so just under half of nests hatched with all siblings within 24 hours, and that's from 18 studies. The range is about 1 to 77 days for all studies, but the mean, if we look across studies, the mean of that is 1 to 17 days. So on average, it took between 1 and 17 days to get everybody out of the nests. And then finally, for the 2 studies I mentioned, we had 12 minutes and 218 minutes as intervals between, average intervals between emergent hatchlings. And so from this, we can see emergence from these 16 species was not particularly synchronous when we define synchronous as within 24 hours. And certainly 2 or more siblings would not be simultaneously visible or detectable by a predator. Okay, so that says something about predator dilution. There were no clear patterns, even though the sample size is not huge. There were no clear patterns between, say, terrestrial, freshwater, sea turtles, but there was a bit of a pattern with the proportion of emerging hatchlings within 24 hours was high in pig-nosed turtles and soft-shell turtles. And that may just be related to a nice sandy nest that's easy to nest in, easy to emerge from. So what do we say then about our hypotheses, our adaptive hypotheses for why we might see synchronous hatching and emergence? Well, from these data is emergence in turtles, other than sea turtles, is not generally synchronous enough to dilute predation risk. So we can kind of X this out as a generality. Maybe things like Podocnemus expansa with large nests and synchronous emergence could dilute predators. The social facilitation hypothesis seems unlikely to me that this would work for your standard turtle nest, shallow nest with a small clutch size. Again, it could be that things like Podocnemus do use this, and the data in this study were not designed to test that hypothesis. So I've put an X with an asterisk there. The third one I think holds promise. I think we can't, with these data, reject the predator preclusion hypothesis. Remember, that's the one where hatching smells are released with the first hatching. And if everybody gets out, it won't attract, it'll attract less predators to the nest. So this one, I think there's a lot of studies out there now looking at what cues raccoons, animals like raccoons and foxes are using to find turtle nests. I think we can add this to that and look at whether or not the synchronicity of hatching and emergence in turtles matches the sort of abilities of predators to detect and destroy turtle nests. So I think that's the fertile area for research in this area. And so I've given that a question mark. And then finally, it's highly likely that very small turtles with small eggs, small clutch sizes, that there will be a degree of synchronicity in hatching and emergence without invoking an adaptive hypothesis. Now, some critics might say, well, Sean, we never thought that there was synchronous emergence in other turtles besides sea turtles. You're raising a straw man. But there's a number of a spate of studies that have not only mentioned that synchronous hatching and emergence is common, widespread, and even ancestral, but they've used predator swamping as the context to set their studies into. So it's not me that's raised any straw man. If there is a straw man, it's the collective of these studies that have raised it. But one of the things that comes up as a problem, issue, if you will, is the fact that we lack a good definition of synchronous. So I think we need a new definition of synchronous. So it's often not carefully defined in most of those studies, even the ones that address predator dilution. Some researchers, including myself, have arbitrarily defined it as 24 hours. Whether or not that's particularly useful, I don't know. Certainly not with predator dilution, that's too conservative. Spencer and Jansen have created a definition for synchronous hatching that is understandable given what they were chasing in the laboratory. But I think it's getting so far from what synchronous really means that we need a new definition and perhaps some new terminology going forward. The other thing we need going forward are getting game cameras on turtle nests. I would have thought that this would be happening at a higher rate than it is. Then we can get accurate emergence data. The cameras are down to $50, $100, so this is really doable stuff. So hopefully this data set will grow in the near future. And then again, I think the predator preclusion hypothesis is the fertile area for understanding synchronous hatching emergence in the field and its and the fit of adaptive hypotheses to that, to those parameters. Okay, that's it for me. Happy to take any questions. Quick plug. There is a chapter in our book that's coming out next year on social lives of reptiles. There's a chapter on synchronous hatching emergence in reptiles. So if you find this area interesting, that'll be perhaps a reason to get the book. You can pre-order it now on Amazon. So I'll try to wait here and see if we get any questions.
There should be some on the way. Just watch your chat, Sean.
Yeah, I'm having a little trouble getting there it is. All right, stack of questions. Here we go. Question 1, apologies if you've answered this already. What did you do to stimulate the vibration cue? Well, initially we discovered it was handling, and then we did some lab experiments where we put them on lab shakers. Pretty crude, but it sort of did the trick. And there's a whole sort of, there's a lot of work in this with amphibians, and my lab's doing it with reptiles, trying to find the right way to vibrate an egg, in many cases to simulate predation, but you know, it's tough to find the right cue when you don't have an actual predator. With the amphibians, with the frogs, Karen Workington's lab uses actual predators, which is the best thing. Question 2, Ricky Spencer. Smaller turtles lay closer to the surface, so any layering will have significant temperature differentials, thermal gradients. It's a gray area. So, you know, the review I did was with all reptiles. And so dealing with small skinks, for example, there's not any significant differences between two small skink eggs that are sitting next to one another. And there's a continuum from that going right up to a sea turtle nest. So when you say significant differential thermal gradients, we don't know where along that continuum it suddenly becomes significant, and that we would expect something like a catch-up that you showed in the lab with a moderate sized turtle. How am I doing for time, guys?
You're great. Take your time.
Okay. Question 3, how might animals that lay satellite nests fall into the context of this? Would the main chamber perhaps synchronize differently from other chambers? Haven't thought about the triple nesters. The satellite nests tend to be shallower. So yeah, it's a tough one, but they only tend to put one egg usually in the side chambers. So not sure how to answer that one. Question 4, Ricky again. Experimental protocols different in many of the studies, some touching, some not. They may take cues throughout development to hatch synchronously and emerge quicker. Yep, so wasn't really in the scope of my review to get into that stuff, and it's fascinating. So I'm just focusing on the emergence from the field, but good point on the mechanisms, and I hope you guys crack it. Another question from Ricky. What about internest communication and synchronous development? Yeah, a possibility. I think for most turtles, it's not an issue, but could be for certainly species that lay communally. Okay, I think that's it. Thank you for your time and thanks to TSA.
Thank you, Sean. Our next presenter has not made it in yet, and so we are going to bring Joel on in when he is ready here in a minute. And if Patrick shows up, we will shuffle him back into the deck. Hi, Joel.
Hi, how's everything?
Doing great, doing great. I'm going to go ahead and step out and let you take the wheel.
All righty. Wait a moment. Okay, everything's good? Okay, so good afternoon. My name is Joel Rojas, and today I'm going to be showing the results of an update made in the status of Terrapin turtles on Hispaniola. First, let's talk about Emiridae. Emiridae is one of the largest family of freshwater turtles that can be found in North, Central, and South America, as well as the Caribbean and other two. And in Europe, you can find two genus of this family. This family has over 50 species that are included within 10 genera. Trachemis, which is one of the genus that we can find in this family, has over 15 species, all native to the Americas. But how do we know what is a trachemid turtle? Well, a trachemid turtle adult may show a rugose notch and serrate posteriorly carapace. Sometimes they may come with a nail. A relatively large blood strand is also present in these species. And in some of them, the males may show longer nails and tails, and also show some signs of melanism. So what is the background that we have of these turtles in the literature? Well, the description of trachemid turtles can be found and their characteristics and diagnosis in Citizen Ernst in both 2012 and 2017. About the taxonomy of trachemid turtles in the Caribbean, we have the early registers by Barber and Carr in 1940, and some taxonomical notes, as well as how the transition between Pseudemis, Chrysemis, and Trachemis was done throughout the years by Settle and Smith in 1986 and Settle by 1988. Uwe and Havas also worked very well in the taxonomy of this group, and Parham with other collaborators work as well in the phylogeny and taxonomy of this group using mitochondrial and nuclear DNA. About the Hispaniolan trachemid taxonomy, Settle and Chelsea made the first status of these turtles here on the island. And in the Natural History Park in Chelsea in 1976 made a monography about the two local species of trachemid turtles that we can find over here. In the conservation part, Powell made several works throughout the years about the conservation status of these species. So which was our goal? Our goal was to gather available information on the distribution, ecological aspects, and conservation status of the genus at the island. For this, we review past and current literature, as well as historical records and online and physical museum collections. Citizen science platforms such as iNaturalist were also used, and field service data was also consulted for this. In the conservation status part, we reviewed the IUCN website, the Dominican Republic's red list of endangered and protected species, both from 2011 and 2018, and current work being done in this area. And these are all results. So we can find here at Hispaniola 3 species of trachemid turtles: Trachemis decorata, Trachemis tenegerivicina, and Trachemis scripta elegans. Trachemis decorata, which is in the vulnerable state, we can find it in Dominican Republic and Haiti. Is a geographical range, is endemic to the island, and we can find in the Caribbean. And as you can see in the synonyms, you can see the changes over the years from Pseudemis to Chrysemis and now to Trachemis. Trachemis tenegerivicina is another species of endemic turtles in the island. You can find it in the Dominican Republic and Haiti, and it's in the near threatened status. And we have the exotic invasive Trachemys scripta elegans. As you may know, this species is distributed worldwide and is an invasive species in most places. Its natural distribution is represented in the south central United States from the Mississippi Valley to Illinois to the Gulf of Mexico. And it has been introduced in several countries worldwide. Here in the Caribbean, it is registered in 2 years, its introduction, and in the Dominican Republic, it was first introduced into occurrence in the 1960s, and the first reported nesting was in 2005. So what do we know about the ecology of this group? Well, there's little ecological information available right now. There's only one work made by Henderson and Powell in 1999 that talks about the ecology of this group in the island. But their main ecological features are similar to other trachemid species found in near islands like Jamaica in this work by Tuberby, Bulbani, and Borjok and Bua. The general information about the Hispaniolan species can be found by Schwartz and Henderson in 1991 and Henderson and Powell in 2009, as well as file surveys and camera traps. And in the Dominican Republic, as you saw before, Sixto Inchauste and Miranda make a very good work with the natural history of this group and also in the ecology part. In the distribution, we can find that Trachemys ecorata is known as the Hispaniolan South Paleo Island slider, and Stenegeri is called as well the Hispaniolan North Paleo Island slider. Around 10,000 years ago, the island was divided into 2 paleo islands and they got united. So here in the island, there is a pattern that were in several taxonomic groups where you can find 2 species, one that is represented in the North Paleo Island and the other one represented in the South Paleo Island. And trachemid turtles are not the exceptions for this. And in the population part, there is no published data by the moment. In the courtship part, we can see that they have a titillation process where the male individual goes in front of the female and start waving the waters with their nails. And this courtship pattern is similar to the description of other trachemid turtles. And there is also a sexual dimorphism where the males are smaller than females. In the mating has been observed between January, February, and August in captivity. In the natural environment, we don't have records right now of the mating season, but in the native status, we can see that the nesting season goes from April to July of each year. And the hatchlings may have a size of 30 to 40 millimeters in the carapace and 26 to 39 millimeters in the plastron. In the clutch, we can see that Trachemys ecorata has around 6 to 18 eggs by Inchauste in 1976 and 6 to 8 eggs by Schwartz and Henderson in 1991. Pablo Felix in recent years has found that the size is around 25 to 45 millimeters. And Trachemys stenegeri has a size around the same amount so the both local species have like the same size in the clutch. And melanism has been documented and is very common in Trachemys stenegeri. It has not been documented by this moment in Trachemys ecorata, and Trachemys scripta elegans in some records have melanism. So here's the current distribution pattern of this species in the island. As you can see, Trachemys ecorata is found mostly in the southern parts of the Dominican Republic, which is this eastern part, and in Haiti, mostly in the central and southern parts of the country. Trachemys stenegeri can be found in the northern and eastern parts of the Dominican Republic, as you can see, and it has a wider distribution than the other sliders that we can find in the island. And Trachemys scripta elegans has been introduced mostly in the eastern part of the Dominican Republic, but we know that it has a wider distribution in captivity. And we can find also a non-feral distribution site in the capital of the Dominican Republic, where you can see over here. So where we can find these turtles? We can find it in ponds around the island, wetlands, and lagoons and lakes. The ponds that you are seeing in the screen are from the eastern part of the Dominican Republic in Bahia. The wetlands can be found in any part of the island, as you can see here, one of Santiago. And lagoons and lakes can be found in some parts of the island, as you can see here, one from Laguna de Bávaro and Laguna de Oviedo. And the conservation status, we have seen that globally, it has been classified as vulnerable from 1996 to right now, the work that is being done in 2020. And in the national list of the Dominican Republic, we can see that it has gone from vulnerable to endangered in a space of only 7 years. Trachemia vicina has been treated as lower risk, near threatened from 1996, and also in the current work that is in process in 2020. And in the national list of the Dominican Republic, we see that it's in the vulnerable state. Trachemia scripta elegans is least concerned since the 1996. As we can see here, the threats that we can find on these species are mostly habitat destruction and climate change, and nest and hatchling predators. In the habitat destruction and climate change, we see that here in the pictures, we can see Laguna del Rincón. Laguna del Rincón actually right now passed from a severe drought very recently, like a month ago. Right now it has more water since it passed a storm Isaias, through the island, but it caused a severe impact in the populations of Trachemia secorata. We suspect that maybe we have lost probably hundreds of individuals in this process. But as well, in other parts of the island, the habitat destruction and climate change may have a severe impact in these populations. About the predators that we can find in the nest and hatchling, we can see that mungoes is one of the most threats, as it is an introduced species in this environment. First, in the nest comes the mongoose. They dig and they take the eggs, and then comes feral cats from local people in the community, and also take eggs of these nets. We can also find rats and unidentified herons in these nets. Okay. In the hunting process also affects these populations. Most hunting happens during nesting season and is done by funnel traps that are called here nasas. Also, people use trained dogs to catch nestling females. And there is also a subsistence illegal hunting happening in the whole island. And these huntings are sold in illegal local markets that are throughout the country. And finally, we can see competition being done by the introduced species, Trachemia scripta elegans, and possible hybridization with local species. The only known hybrid that we have over here is the hybrid between the two local species, Trachemia secorata and Trachemia vicina. It has been documented from 1976 up to the work that worked entirely on the hybridization process on these turtles in 2013 by Parham and their collaborators. What is the current work that we're doing here in the island? Well, in Haiti side, Jeffrey Paul is working with the current populations of Trachemid turtles, specifically Trachemia secorata, in Haiti's side of the island. In the southern part of the Dominican Republic, we have Pablo Felix working in the nesting process of Trachemia secorata as well. In the capital of the Dominican Republic, myself, I'm working with the hybridization process in this non-feral distribution site, where you can find the 3 species of turtles here in the island. And Joan Cabrera is working in the population structure of Trachemia vicina in the eastern part of the Dominican Republic, in Laguna de Bávaro. And in the whole island, there is also the current work being done about the conservation status of these species in the island. So what is being done right now as well? In the legislation part, there are two laws that protect these turtles. There is the General Environmental Law and the Biodiversity Law in the Dominican Republic. And these species also are found included in some protected areas in the island. Trachemia secorata can be found in Laguna de Cabral Wildlife Sanctuary and in two national parks, Lago Enriquillo and Jaragua. Trachemia senegeri can be found in three national parks, Humedales de los Ama, Los Haitises, and Montecristi, and in the Wildlife Sanctuary of Laguna de Bávaro. And so what are the conservation actions that we think are needed in order to protect these species? Well, we need more research in the population and ecological status, potential impact of climate change, genetic studies of potential hybrids, and natural genetic variability in the native population, exit conservation programs to establish assurance colonies, removal programs of Trachemia senegeri vicina from wild and easy to escape, and citizen education on the negative impact to release turtles of pet turtles, because that's a common practice here in the island to release pet turtles into the wild, and they can cause a severe impact in the genetic stability and also in the population stability of these turtles. We would like to acknowledge Janet Mateo for reviewing this work, as well as Jeffrey Paul, who gave us information about Haiti side of information, as well as TSA and IUCN for the invitation to present this. And these are the credits for the photos. This is the literature that we have reviewed throughout this work. And if you would like to contact me or Fixing Chelsea, here are my contacts and my social media. So thank you very much. And let's see if there is any questions. Yes, there are several questions being done. Okay. Look for the first one. So the first one is from Cameras and says, what is the salinity of some of these wetland habitats? Can they handle more saline environments? Well, in Laguna de Cabral and Laguna de Oviedo, where you can find it in the southern parts, the salinity is very high. Right now, I don't have the number, but it can go approximately from 75 to 80 parts per 100. So they can be found in very saline environments here in the island, as well as others that are not that saline. So we have the question number 2 is Joel, with recent deaths of Trachemia secorata in Laguna Rincon, do you think the status of should be elevated to endangered in the IUCN red list or the national list of the Dominican Republic? Well, we will need to make population assessment in order to increase or decrease the status of the status in the conservation part. We will need to see if the population was severely impacted, because that is, and I didn't say that in the presentation, I'm sorry, but this is the only, probably the only natural and like pure population of Trachemia secorata that we have in Dominican Republic side. So yes, we will need to verify the population status right now in order to verify if they if the status can be elevated to any other location, like endangered or critically endangered. Jura Riley says, hi Joel, thank you for the great talk. I love hearing about your work. In your opinion, how significant is the threat of illegal hunting to the turtle populations? That's a very good question. Actually, here in the Dominican Republic side, the problem is there, but it has not. It's not that big as in, for example, Haiti side. But we also have the problem of not only illegal hunting for selling them, but we have also the practice for magical or religious practice that people think that they're aphrodisiacs in order to use them for virility or to cure even coronavirus. In the recent days, we have a news that the blood of turtles cure coronavirus, and one person injected them and it was taken into the OR. So yes, there is a threat of illegal hunting, and I think it can be big, but we need to make more studies about the real impact of illegal hunting in these populations. And I don't know if there are more questions. Okay, so we think that's it. So thank you very much for everyone to listen to this conference, and I'm gonna give the microphone to the next person.
Can everyone hear me?
We can.
All right, can everyone see my screen?
Yes.
Okay. All right, so we'll get started. My name is Matt Cross, and today I'm talking to you about some of the research that we've been conducting here with our partners. This is a very large project that involves a lot of people. Most of them are named here. It's really kind of a mouthful, so we just kind of like wrapped ourselves up under this generic Blandings turtle working group. It's not really anything official, mostly just trying out the logo for size. So we'll go on and get started here. So when we talk about Blandings turtles, if you're not familiar with Blandings turtles, that's a conversation I suppose we can have at a different time. But they're a medium small aquatic turtle, about the size of a American football when they're full grown. And their range kind of is here in the Midwestern Eastern United States. Throughout their range, there have been a decent number of surveys conducted, but most notably, Ohio and Michigan kind of fell behind. So we know when take for Michigan, for example, there are a lot of element occurrences. They've had more than 200, but to the best of my knowledge, the only long-term study there is the E.S. George Reserve. A lot of other little side projects have been undertaken, but nothing at the any kind of thorough survey. The same thing in Ohio. We've had a few offshoots. We've had a long-term survey here from the Toledo Zoo in Northwest Ohio for about 15 years. Same thing in a few places elsewhere, but not really these thorough assessments we've seen in these other states. And so this became really important as the Blandings turtles are on the list for the candidate list for listing on the Federal Endangered Species Act. And so we want to have as much information for the to pass off and to help that listing. Sorry, I'm trying to multitask here. And there you go. So our group got together. We set out to we applied for a competitive state wildlife grant, and our goal was to survey the status of the assessment, or do a status assessment survey for Blandings turtles in Northwest Ohio and Southern Michigan. This group, we're the ones who applied for the grant, but then our technicians, they're the ones who actually did all the work. They're the boots on the ground. We had quite a few people involved in this, and it was a pretty awesome effort. Our technicians were really great, and they also took plenty of time to conduct side research, mostly with Oreos. So our project had four main objectives, mainly to develop and implement a monitoring strategy, and not necessarily develop but modify the monitoring strategy of the Northeast Blandings Turtle Working Group to fit our needs. We're going to create distribution models to prioritize areas throughout the range, our study area. We gathered a lot of genetic samples to look at that aspect of things, and we really wanted to develop a unique conservation plan for our sites that would be beneficial to the states moving forward. Today, I'm mainly going to focus on the implementation of the monitoring strategy, the coordinated monitoring assessment. We could be here the rest of the day talking about these other things. So we'll kind of touch on them, but the main part here is we're going to focus on the surveys. So this was, again, we used the conservation assessment methods from the Northeast Blandings Turtle Working Group. This is a two-tiered approach that focuses on long-term intensive surveys as well as rapid assessments. These take place in wetlands, have different focal areas, and are designed to answer a couple different questions, mostly on where they are, the long-term sites, kind of get into those population numbers at those individual locations. And so this two-tiered approach worked out really well for them. So we just kind of modified it for our uses. and it worked out pretty well. Our study area, again, we're not going to really focus in on the details here. We picked the Lake Erie watershed because this kind of gave us a boundary in Michigan. So the Lake Erie watershed really encompasses all known Blanding's turtle locations in Ohio. Michigan, as I said, they have more than 200 element occurrences in almost all the counties in the Lower Peninsula. So we wanted to pick a reasonable boundary for our survey, so we weren't trying to survey the whole state. So we confined again to the Lake Erie watershed, and we were sites in Ohio and Michigan. The habitats we were in ranged from the pristine, Lake Erie wetlands that are very large and unfragmented to these early season flooded forests. And in a lot of cases, especially in Northwest Ohio, ditches. The habitat use in our end of their range varies greatly, and it can kind of run the gamut. So the surveys, it's not popping up. Anyway, let me see here. So our surveys, we used hoop and Promar traps, and it involved a series of 4 focal points. Each focal point had 5 trapping locations. The trapping locations had, each trap location had 1 hoop trap and 2 Promars associated with it. They were baited with trout and fish oil, and these were placed throughout wetlands and allowed us to look at the kind of distribution throughout those areas. Honestly, putting the fish in the fish oil was the least favorite part of our technicians' jobs, but they got used to it. So honestly, we can just dive into the results. Again, I apologize that one picture wasn't showing, but kind of give you an idea of the framework of where the trapping strategy within the wetlands. So in Michigan and Ohio in 2019, in Ohio, we were able to survey 37 sites. In Michigan, 29. We put in a massive amount of effort in terms of trap nights for these turtles. For the abbreviations over there, EMBL are going to be Blanding's turtles, CHPI, painted turtles, CHSE are snapping turtles. And we had a pretty successful first year. In total, we put in over 12,000 trap nights. And as you can kind of see, our Blanding's turtles per trap night varied between Michigan and Ohio. When we look at comparing this to the data from the Northeast Blanding's Turtle Working Group, we can kind of start, since we collected these data in the same fashion they did, we can start looking at these and looking at this from a bigger picture. So we put in a pretty comparable amount of effort, and we can look at these Blanding's turtles per trap night, but I tend to lean towards trap nights per Blanding's turtle because it's easier for me to visualize. And honestly, so we needed more trap nights in Ohio to get a Blanding's turtle, but that really varied greatly throughout the trap locations. So we can see here that it ranged all the way from 1 to 2 trap nights to close to 250. The OSU crew put in a lot of work for this. In Michigan, MNFI, it varied again. And so there's a lot of factors that go into these different sites that kind of tell, how easy it is to catch the turtles. Again, our total turtles captured for all 3 of those main species fluctuated throughout the year, but we had a peak in late May. So we looked at total captures and captures per trap night. Really towards the end of May, there was our witching hour or witching week, and then it kind of died down throughout the rest of the season. Although Michigan did have some luck in late September, which was pretty cool. This is just fun. If we start looking at how the variation within the weeks, this is just from one of our sites, and it just really, really crazy and really interesting to see how much variation you have at the same location. Our sex ratios, where we were encountering Blanding's turtles, were pretty even male to female, depending on where you are, at least in the places where we got a decent number of turtles. And our age structure did tend to be more adult skewed, but that's kind of to be expected. There were several sites where we did get some of the representatives from all size and age classes in our traps, which is great news. We're able to kind of compare preliminary. I'm still working on teasing this out. Are the hoops versus Promar traps in terms of turtles per trap night. And for Blanding's turtles, it was pretty even. For painted turtles, you know, slightly more so for hoop, and for snapping turtles, definitely, you know, leaning more towards those hoop traps. One of the really cool parts about this was our community science. We've had a long-term volunteer who gets his community involved in reporting turtle sightings for us, and we've rolled this into our surveys. So he also does visual surveys for us, and then I have a teenager who does visual surveys near her house. And so even though 2020 was kind of a wash for a lot of us, we were able to keep the the community science project going and got some really cool data. We did an outreach program where we handed out this. It's a magnetic leave behind, so it's got all our information on where they can report Blanding's turtle sightings. And this actually generated, I think, 5 to 6 new potential sites. 3 of them confirmed, and 5 to 6 sites early on their potential. 3 of them have confirmed with the Blanding's turtles, and so this is a pretty successful effort. And while we're out in the field, we had so much time to interact with the public and really to spread the word and get people interested in this. We entered a photo competition with Memphis Net and Twine. We did a video here kind of showing what we do from the Toledo Zoo, and we had about 12,500 views. So we really hammered home the education outreach side of this, along with the main part of our grant. Really, we're not just collecting data on Blanding's turtles, we're getting data on snapping turtles and painted turtles. So if we look here, our current harvestable limit in Ohio, if we increase that by 1 inch, we actually end up protecting about 80% of our potentially harvested females. And just for fun, Conan 1951, his maximum straight carapace length was somewhere there in the 330 range, and we definitely knocked that out of the park. So in 2020, it was kind of a wash, but we did we updated our distribution models and revisited some of our sites. Our geneticists kind of started doing their work, and we've got a couple of grad students working on the landscape genetics as well as the occupancy modeling. In year 3, we're going to wrap up our surveys. We're planning on revisiting some of our sites, kind of testing out some new tweaks to the methodology. And then we'll be working on that conservation plan. We've already started talking with Midwest Park and had discussions with the USDA NRCS, the Working Lands for Wildlife group. And yeah, so I think that with that, I just want to acknowledge all of my helpers, big and small. And I will take any questions if I can figure out how to find the Q&A bar here on my thing. There we go. Yeah. All right, so we had a couple of questions early on. Do you have evidence for interspecific competition affecting Blanding's turtle numbers? Not really. I don't have any evidence of that with the data we have, but that's one of the things that I think we can hopefully look at with some of this multi-species, this multi-species occupancy stuff is, maybe does the presence of one affect the other? I doubt it. I think there've been a lot of places that have worked on similar things like that. But yeah, no, we haven't seen any evidence. And then were sex ratios and the trap types very different, more male bias and hoop? So I've read a few papers on this. I haven't looked at this in our data yet. I would tend to lean towards yes. In general, I think basking traps tend to favor more of the females, whereas the hoop traps tend to be more male biased. However, if I remember correctly, we didn't see that in the Blanding's turtles. It was pretty even across the board, especially with some of our long-term data sets. Let's see here. Any other questions I missed? Question 1, question 2. All right, if there aren't any more questions, I guess I can turn it over to the next person. Oh, question 3, sorry. How do you know you're not recording the same turtle multiple times? So they're all marked. So that each one, we spent a lot of time marking every individual turtle. So in some cases we do recapture them, but in general, at the rapid assessment sites that are only a 4-day survey period, we don't get a lot of recaptures within that time frame. But yeah, so all of these turtles are processed. The Blandings turtles, they additionally get pit tags in addition to the unique shell notching codes. Okay. So we have one more question. What is the conclusion about the status of the turtle in Ohio and Michigan and the Lake Erie watershed? So it depends on where you are. So in general, we had pretty decent success, especially within you know, some of our better sites. Same thing in Michigan. However, once you start to get in some of these other parts of Ohio, especially where the Great Black Swamp used to be, the numbers start to decline. So it's kind of one of those things where they're doing well, where they're not, they probably aren't. No. So very last question. We only caught free red-eared sliders, and so I didn't present those in the data. All right, thank you everyone, and we'll talk to everyone later.
Hola. Laura, ¿cómo está? Sí, muy bien, gracias. ¿Qué pasó? Vamos.
Okay, hi everybody. My name is Tracy Castellon, and Pepe, I'm on air. Okay, I'm sorry. This is Tracy Castellon, and I work for Florida Fish and Wildlife Conservation Commission. I'm in the research branch, and we are engaged with a status assessment for the diamondback terrapin. This is very much a joint effort with our partners. We are working with essentially everyone who is interested in diamondback terrapins in the state of Florida. And so I will acknowledge those people to the extent that I can. Assessing diamondback terrapins in Florida is important because we have significant regional responsibility for the species. We have about 20% of the species range because of the long linear extent of the Florida coast. We also have 5 of 7 subspecies, and 3 of those are endemic. In Florida, we use the IUCN Red List criteria for all of our assessments. And so this project will focus on those criteria that are most likely to be triggered by the diamondback terrapin. Those include a look at the geographic range reduction, population, small populations or restricted populations, population size reductions, and then declining trends. Also, one could look at the quantitative analysis of population models, but at this time, we just don't have enough data to do that. So we're hoping to move into that kind of analysis later and once we have more data. But right now, we're focusing on the others. And so our research plan essentially follows the IUCN criteria. So our first criterion is to obtain sighting data to map the current distribution. That will help us determine if there's evidence for a range reduction. We're also looking at developing a habitat model using those those sightings. We're collecting tissue samples and conducting a genetic analysis, which will help to validate the subspecies classifications, because meeting those IUCN criteria is very much related to the population that you apply them to. And we're also estimating effective population size and isolation where those issues are important for conservation status. We're also conducting mark recapture studies, and we're working with partners who are collecting their own data. We are estimating population declines based on past and predicted habitat losses and degradation, because there's very little trend data on terrapins in Florida. And so we have to look at habitat loss kind of as a proxy for population losses. I'm going to present the methods and the progress to date together, because this is still very much an ongoing project, and I think it'll make more sense to present it that way. So the first criterion that we looked at is the range reduction. We collected over 10,000 sightings from our partners statewide. The red lines indicate where we know terrapins are currently distributed. These are sightings from the year 2000 and onward. The historic data are the yellow lines, and this doesn't indicate an actual range expansion. It's just that we had very little data before this project. So in short, it looks like there's no reason to expect a range reduction. That doesn't mean that there haven't been populations that have winked out. They probably have. But for the time being, there's no indication that this criterion for listing or for conservation status is going to be met. Now, the population size and trend criterion relates primarily to very small and isolated populations. As I mentioned, we have 5 subspecies in Florida. The Mississippi diamondback terrapin, which is just here and just reaches its eastern extreme in Florida. The ornate diamondback terrapin that covers most of the Gulf Coast. The mangrove terrapin in South Florida. This is a very uncertain boundary. There may be, it may be that the population of mangrove terrapins is limited to the far southern Keys. The East Coast terrapin covers most of the East Coast, and then we have the break with the Carolina terrapin that extends further north of Florida. So if this criterion is met, it would probably be for the Mississippi diamondback terrapin because it has such a small range in Florida. And then possibly for the mangrove terrapin, if it is in fact limited to the southern Keys. But as I kind of hinted, we're not 100% certain about the classification of these subspecies. There's evidence that, that the species may be oversplit. Their recent genetic data don't support that level of differentiation. And in fact, the very most recent paper by Kristen Hart suggests that these 4 subspecies are probably should be combined into a single management unit. But those studies were done based on a limited geographic coverage of data, and then there were microsatellite studies, so they had limited genetic coverage. So what's needed to really address this question is a larger, more geographically extensive coverage and using next generation genetic techniques. So that's the next part of our study is we've partnered with some genuine geneticists, Brad Schaefer, who's a world-renowned turtle geneticist, and his colleague, Peter Scott. And this project will assess the current and recognized subspecies with the goal of combining some of them if that's warranted, and then identifying any truly genetically distinct management units. We're also estimating effective population sizes because that will inform some of our statewide assessments. And we're measuring genetic isolation where that's important for conservation, particularly in the lower Keys. where that population may be extremely isolated. The methods we'll be using are whole genome sequencing and rad sequencing. We'll use whole genome sequencing for the subspecies question, and then population genetics will be, whole genome sequences for the subspecies question, and then rad sequencing for the population genetics. So that those together will be the most exhaustive molecular approach that's currently available. We want to do this in a way that's very defensible because it may be controversial. We want to use a method that will identify any genuine differences that can be used to conserve the subspecies. These are the data we've collected so far in terms of genetic tissue samples. We've collected over 1,000 samples in Florida, and we have pretty good coverage geographically. There are some areas that where we don't have good coverage that are important because they are at the boundaries of subspecies. So we really would like to fill those gaps. We're reaching out to people. If you are aware of anyone who has samples from these areas or could point us in the direction of a surefire place to get turtles, we would very much like to fill those gaps. For our range-wide genetic samples, this is what we have so far. We have some big gaps here. We had plans this season working with partners to fill those gaps, but because of the coronavirus, a lot of people are on travel restrictions and a lot of people haven't been in the field. So we will be scrambling in the next year to try and fill these gaps. If anyone can help with that, please do reach out to us. These are the areas where we have population, we have enough samples to run population level genetics. The red circles are places where that work is complete, and the yellow circles are places where it's ongoing, but where we anticipate having enough samples. These are the areas, aside from the genetic work, we're doing actual in the field mark recapture and monitoring studies. The yellow circles are the places where Florida Fish and Wildlife has conducted studies, mark recapture studies. And then the red circles are all of our partners who are sharing data with us throughout the state of Florida. And this is very, very helpful. We're going to know a lot more about the status of diamondback terrapins in the state of Florida when this is finished. So all these data are going to be informing the population size reduction criteria. We don't have a lot of trend data in Florida. All that we have in terms of direct observation so far are Kristen Hart's data on the Western Everglades. She has seen a recent crash in her long-term monitoring site. In the Central East Coast, Siegel monitored a population that was fairly robust in the 80s and then found when he revisited in the 90s that it had essentially crashed. They're still present, but at very low abundance. We have our two study areas in South Florida, where we are following up and resurveying the same locations that were surveyed by Brian Mealy and his colleagues over a period from the mid-90s to 2010. So we should be able to identify trends there if they exist. And then we have a partner in North Florida who's been monitoring an important nesting site for well over a decade, and we should be able to find trends there. Where we don't have trend data, we're looking at habitat loss from urbanization, coastal development, shoreline hardening, and also accounting for sea level rise. This will allow us to predict into the future whether we anticipate triggering the IUCN criteria based on habitat loss. So that concludes my presentation. We are still seeking partnerships, and in particular related to tissue sampling. and especially from people outside the state of Florida. So please do get in contact with us if you have any suggestions on how we might fill those gaps, because this is something I think that's going to be beneficial to diamondback terrapin conservation range-wide. And I'd like to thank our partners. There's too many to list individually, but you know who you are, and we very, very much appreciate your efforts. I'm more of a puppet master, and most of the work, most of the on-the-ground work is coming from our partners, and we appreciate it. And with that, I'll close, and I'll take any questions. I can't get the chat feature open. Does anyone see any questions coming in?
Give us just a second, Tracy, and we will get those to you. All right. Where did you get your funding for genetic work? Is it through the state?
It's a state wildlife grant, so it's federal money that comes to the state, and then the state distributes it based on a proposal process.
Second question is, are you located at the Gainesville Research Lab?
I am located in my upstairs bedroom in Orlando. But yes, my lab is the Gainesville lab.
We'll wait another minute or two to see if a couple more come through. And I think those two may have been it. Thank you very much, Tracy.
Thank you.
Okay. All right, here we go. Are we good? I think we're good, David, yes? So first of all, I am not Gamaliel Castañeda Gaitan. I'm Craig Stanford, and I'm one of the co-authors presenting for our research group today. So I want to begin by acknowledging Gama, who has been studying the species Terrapin quahila for almost a decade now, and the other co-authors on this paper are his junior colleagues and graduate students, and all of them working for years on this project. I've been working with them for the past 3 years, and it's really been a pleasure and an honor to be able to be with this really good group who are from the Universidad Juárez del Estado de Durango. So my paper is on the conservation status and sex ratios of the Coahuila box turtle in the Cuatro Cienegas Valley of Coahuila, Mexico, during the past number of years. So first of all, I'm guessing that many of you know something about this species. Those of you who are from North America or people in general are very familiar with the genus and with box turtles. And you probably know that this is a very divergent member of that genus. It is often called the aquatic box turtle, not because it is entirely aquatic, but it is largely aquatic with the ability to make use of terrestrial and aquatic habitats both. You may also be aware that it has an extraordinarily restricted range in the state of Coahuila in northeastern Mexico. And that is its primary problem, and that we'll be talking about as I go through this talk. Coahuila box turtle is on our list, on our specialist group, our IUCN list of one of the 25 most threatened turtles in the world. And it belongs there because not only are the numbers small, as you'll see, but the threats are many and increasing. So the area I'm going to be talking about today is the Cuatro Cienegas protected area, which is basically encompasses this entire area, the majority of this valley, which is kind of an M-shaped or maybe W-shaped valley with the mountain range that bisects it. And this map has been published now many times over the past 35 or more years or 40 years with regard to the turtles in that valley. The distribution of the turtles has not changed except that the densities have changed, as you'll see. And so I'm not really showing you anything brand new here. And the topography of the valley is enormously important when we talk about the threats and the possibilities that the animal has for future survival and any management plan for them. So I'll show you just a few pretty images now. The valley is a very beautiful place, and it is best known not only for turtle biologists, but for aquatic biologists, for ecologists, for microbiologists, for ichthyologists, as a place of a very high level of endemism. There is permanent water in the form of these wetlands. These wetlands are fed both by aquifer under the ground and also from runoff from those nearby mountains. But in addition to the permanent water, there is an enormous resource of ephemeral pools, and those have been the resource that has been drying up rapidly over the years in this valley. So there was a time in Cuatro Cienegas Valley during the rainy season, virtually the entire valley was flooded an inch or a few inches deep with water. Those days are long gone, and now there are very large dry areas across which the turtles would have to pass to migrate in order to get from one appropriate habitat to another. It's probably best known, the valley, not really for the turtles, although that's of course our concern. If you look at the lower left-hand image, and there is this shelf of white material all along the banks of this water hole, those are stromatolites, which are colonial microorganisms that they're certainly not freshwater coral, but because I know very little about them, I call them freshwater coral sometimes. And they are often used by people who are studying the evolution of early life on Earth as analogs, because they've been around for a very, very long time. This is one of the best places in the world to find them and see them. And once the wetlands dry up or are degraded, they go away permanently. As you can see, there is this, because of mineral content in many of the bodies of water, there is this spectacular desert landscape. It's in the middle of the Chihuahuan Desert, and it is an arid scrub habitat rather than proper desert. But for practical purposes, it's a very arid place. But then there are hundreds of these pools that are turquoise or green dotting the landscape. Some of them, like the one at lower right, are only a few meters across and are deeper than they are wide in diameter. And this is a habitat for 10 endemic fish species. If all 10 remain, there's a huge problem with invasive fish species in the valley, and for 3 aquatic turtles as well. So before we get into the data on the box turtle, let me mention that there are 4 chelonians in the valley. In addition to box turtles, you have an endemic Trachemes taylori. Gama and I have a master's student who's doing genetic work right now on this species, mainly to see if there has been or how much introgression there has been from red-eared sliders that have been released in the valley. That's a huge genetic threat. For all we know, because we don't have good census estimates, this species may be more threatened than the box turtles are. They are much more reliant on permanent water than the box turtles are. There also is an endemic subspecies of the spiny softshell, Lavapeloni spinifera, that is in the right habitat fairly common, but the question is whether that habitat, which is permanent water, will remain into the future. And in addition to those two, the Cuatro Cienegas Valley is the southernmost point of occurrence for Gopherus berlanderi, and there is some debate about whether this population, which we only find in the on the northern side of the valley in the drier areas, is a natural population or possibly an introduced population from released pets, because it's a disjunct population from 20 or more kilometers to the north, at which point the species is found much more commonly. So these three, in addition to the box turtle, are all found in the valley. And as I said, two of the species are endemic, and one of the subspecies, the one subspecies, is also endemic. So the species, the box turtle, has been studied for a long time. William Brown was a graduate student at Florida State who began a study in the late 60s. And he was really the first person to do a systematic field biology study of Coahuilan box turtle. He documented a high density of animals and an estimate, and it's a pretty rough estimate, of a relatively large population of 10,000. The methods used for all three of these studies are different, so they're not really directly comparable data. Jennifer Howarth at the University of Alabama, beginning when she was a graduate student at UT Austin, did a study of genetics, gene flow, migration, and other aspects of the species. And she, working about 30 years later or so, found very different results and a much lower population estimate. That is possibly due partly to her methods of calculating, estimating densities, but it also is certainly due in part to a rapid transition of the valley from, as I said, a seasonally inundated place to a place with increasingly few and far between reliable aquatic habitat for the turtles that are present all year round. Her population estimate was only 1,500. And just to jump to the bottom line conclusion for our study, Castañeda's work, and this was published, by the way, I meant to say, in the latest issue of Chelonian Conservation and Biology. This paper I'm giving is published in that issue. Castañeda and colleagues found a population estimate overall that's pretty much in the same range as Howarth, if you take an average figure between 600 and 2,200, and densities that vary dramatically depending on which wetland area we're talking about. So this table shows you eight different wetland areas and the densities of turtles recorded in them between 2011 and 2015. And you'll notice it's very obviously that one wetland in particular, which is called Los Gatos, which is pretty much in the dead center of the valley. So it's maximally distant from the town, the pueblo of Cuatro Cienegas, which is rapidly growing, and also from other potential sources of habitat degradation, agriculture, which is present surrounding the valley, and then also from the mountains. Has much higher population density in every estimate that's been done, including up to and including today, in fact, than any of the other wetland areas. And I'll just point out that the last wetland, which I'll show you a photo of in a bit, Churince, essentially no longer exists. So in fact, when Brown did his study in the 60s, early 70s, there were more wetland areas than these present. And some of those are just no longer wetlands there anymore. They've dried up because of a drop in the water table. And we'll get to that in a minute. And Churince is the most recent wetland that no longer exists. It's simply a dry, sandy area. So Castañeda's study was interested in the number of turtles, the sex ratio of the turtles, the age breakdown of turtles in the valley for the Coahuilan box turtle. And what he found was a quite skewed sex ratio in male bias in favor of males, for reasons that I think we really aren't quite sure of today. The other disturbing thing that we found here, in terms of, this is the sex ratio slide, is that of all the turtles that were captured, and the number is 393 turtles captured over the years of the study, of which 13%, 51 animals, were previously marked animals, because each study had notched turtles, that only 2 juveniles have been found by this study in a total of what, 8 years, 9 years, or field seasons. And that, of course, is a great concern. There have been a very large number of nests, turtle nests found of all species, with egg fragments destroyed by predators. And we don't have empirical data on this, but it certainly appears, as the town has grown, that the number of subsidized predators, coyotes, foxes, skunks, and so forth, has grown rapidly, also raccoons. So this is a concern, a male bias sex ratio, and seemingly very little recruitment of juveniles into the population. So Castaneda's team has done a lot of modeling using various variables, from isotherm, elevation, aquifer, and they have come up with a total suitable habitat in the valley indicated in this kind of heat graph, heat image by the dark red coloring, of only 540 hectares of suitable habitat. That represents 0.06% of the total area of the protected area that's outlined there on the image for you. So again, 40 years ago, much of this valley was at least seasonally appropriate habitat for the box turtles, and they ranged far and wide from these areas of permanent or ephemeral water out onto the landscape. And Howarth recorded gene flow in the form of migration of turtles kilometers, and I believe up to 8 kilometers away from one site to another over a period of one season, one field season. So today, the amount of suitable habitat for these animals is dramatically reduced, pretty much corresponding to the loss of water in the valley. So the extreme seasonal changes in water is a natural part of the ecosystem, because during the dry season, a lot of the water would go away, but historically when the rains came, the water sources would not only fill up, but overflow onto the landscape, as I said. So what we see today is partly the natural history of the area, but also greatly exaggerated by a drop in the water table. And the drop in water table has been caused, we don't have data on this, we don't have data on this, but hydrological data indicate that water table drop is corresponding to a growth in agriculture around the valley. And I'm talking about large scale industrial agriculture. Northern Mexico is, as you know, a beef cattle area. All of that carne asada is raised on fodder, on crops, and those crops are what are being grown in large scale agricultural projects that require large quantities of water around the valley. And so even in terms of conservation management, the Turtle Conservancy is greatly involved here, but even in terms of conservation management, it's not enough to acquire and protect the land unless you also acquire and protect the water by acquiring the water rights underneath. And this is a major conservation issue and a major conservation problem going forward. So this is actually Churince. This is a wetland that during Brown's study decades ago was a thriving wetland. And in fact, not long before I joined this study, this was also a thriving wetland. This happens to be located at the extreme southern end of the valley. So it was the most vulnerable place because of its proximity to large agricultural projects that were taking water from the water table for irrigation and thereby dropping the water table. And when I visited this site with Gama 2 years ago, we found skeletal remains of all the 3 species of aquatic turtles. We even found a very recently dead carcass of a Trachemes, but this is today a wasteland, essentially. It's really just become part of the desert, and whether it can be restored in some way in the future is maybe an open question, but obviously not with a great deal of effort and a great deal of funding as well. So this is a wetland that has gone the way that we fear others of those 7 other wetlands I talked about will go if we don't do something about the loss of water from the valley. And finally, the bottom line problem here is also that This is an agricultural economy historically in the Cuatro Cienegas Valley. However, it's undergoing a fairly rapid transition, it appears, to a tourism economy. Not know about Cuatro Cienegas, but it's a well-known resort valley, a vacation spot for people living in northeastern Mexico, coming from Monterrey and other areas. And so at least for a couple of months during our summertime, it becomes crowded with tourists. There are wineries, there are hotels. It's a beautiful place. Some of those posas, some of the water holes that you saw, the turquoise ones, are open to the public for swimming, bathing, water sports. And so there are many tour operators moving into the valley, and kind of a land grab going on in which the land is being bought for various purposes, ranging from speculation and investment to establishing or building a tourism industry. There are rock climbing ventures on the mountains surrounding the valley. There are hiking trails. There is a lot of conversion of the traditional economy, which itself was harmful to the wetland habitat and the turtles because of the growth of these industrial agricultural projects. And now as well, tourism, which in theory could be a good protector of the habitat. But as we all know, it's a double-edged sword in that more development because of natural beauty leads to more people and continued development, often to the detriment of the animals themselves. There's a plan for, there have been plans for golf courses, water parks, a zoo, and so this is a rapidly growing area of development. And I just want to conclude by thanking everyone who's given us funding over the years, and thanking my wonderful colleagues, and thanking our two respective universities as well. So thanks. And if there are questions, I will try to take them.
Craig, you should have a couple in your chat thread.
My chat room here. Not able to access, oops, sorry. Not able to access my, my chat for some reason.
Keep your slideshow up and move your cursor to the top of your screen to look for the
I'm looking for, here, okay.
chats.
Okay, see, no. Are there questions?
I think there are. Give me just a moment and I will.
If you could read them to me, I'll try to handle them.
Yeah.
Oh, okay. Let's see. I can see the questions now, David. So question one, which other box turtle species live closest by distance from the valley? That's a good question. Closest by distance. There are no box turtle species for hundreds of kilometers. I would say many hundreds of kilometers. Mexicana, depending on whether you consider that a subspecies of Carolina or a species, might be the nearest, but it's pretty far away. It's in Sinaloa and those areas. And if you were to go north, this is about a 5-hour drive from the Texas border, from the South Texas border. So if you went north, I suppose you would eventually hit the range of Ornata, but you're quite a good distance away. Another question, has the team found any Brown's marked turtles? I believe the answer is yes, and I'm going to defer and ask anybody who has questions that I can't answer to email Castañeda Gama, who is standing ready to answer those questions, but I believe the answer is yes. Question, any chance of buying wetlands through Nature Conservancy or other? Yes, in fact, this is something that we're all actively interested in. Turtle Conservancy has been involved there. However, the development for tourism has meant that land values have skyrocketed. And so we're talking about land that's being valued by lots of people, including wealthy, powerful interests in Mexico who see the value of potentially developing it. Do I have time for another question? Let's see. What is the current political and sociocultural climate like in regard to conservation efforts? What are efforts to improve this? We work very closely with CONAP, which is the division of the government that works with natural resources, and they are very eager and interested in protecting this area. the delicate balance, kind of the dance, and many people who work with this kind of threatened species situation are familiar with this, is working with local landowners. Most of the land is owned by land collectives, ejidos, and then you have developers, as I said, coming in wanting to buy land and potentially buy land from the ejidos, and the government's kind of in the middle trying to make sure that private interests are doing the right thing with regard to the land and the animals on it. And in my experience, they have been a very much a positive force in the valley. The question really is, with the population, human population increasing rapidly, and with a great desire to buy plots of land, parcels of land, how much can the government do to really regulate and control that process? And if I have time, one more question. With the given threat of climate change and excessive water consumption, do you foresee a point at which there will no longer be habitat for this species? How will this affect conservation? I have to say that I certainly foresee, and I think Gama would fully agree, a future in which the habitat for this species is dramatically reduced further, so that the wetlands that I showed you in the table, in the image, that most of those will no longer be habitable unless something really changes. There are examples around the world of mitigating some of this water table loss through various means. And if you do that, you can actually, in theory, quite rapidly restore a wetland. It takes a lot of work. So there is that potential for the future, but absolutely, if development continues uncontrolled like this, it's going to be a problem. I should mention, there is a wonderful nonprofit working in the valley, Pro Natura Noreste, and they have done some great stuff. There are canals that run through the valley that have been put in for decades to irrigate crops, and they've sucked water away from the valley, and Pro Natura has been involved in actually negotiating and removing some of those canals, which is very good for the water supply to the wetlands habitat. So there is hope. There are many people working on these problems, and many people in the government and the nonprofit community, including us, are really eagerly interested in trying to mitigate the situation in terms of water. It's all about the water. And then last question from Tim Gregory: what can we do? What's the conservation plan? I think that's the answer that I just gave. And about what is the Turtle Conservancy's role? Again, as I said, there is a desire to do that. The best way to protect this land is for nonprofits to work with the local community, not against the local community, but become landholders and land sharers, and really primarily back the Mexican nonprofits who are deeply involved in trying to do the same thing. And again, the problem is that the land is relative to some areas of potential turtle habitat that are in the middle of nowhere. This increasingly is in the middle of somewhere where there's a lot of interest in that land. So the prices are going up rapidly. And that is the last question that I have. So if we're good, I will turn it back to the next speaker. Does that sound like a good plan, David?
Absolutely. Thank you, Craig.
Thank you.
Hi, hello everyone. Let me just open my chat. Right. Okay. I am Isabel Eli. I am talking here from Northern Australia, so pretty much nearly 3 o'clock now, 3 in the morning. I would like to start acknowledging Varai, Kungaranka, and Maranangu people, the traditional owners from the lands where we have been working on. And what I will be sharing with you today, it's a data about the metal assessment of the long-necked turtle Chelodina rugosa, or Chelodina rugosa in English, in the Finniss River floodplain in the northern Australia. And this is my team. So we are working at the Charles Darwin University with some partners. So the long-necked turtle, the Chelodina rugosa, was Chelodina rugosa then changed to oblonga, now it's rugosa again, belongs to the family Chelidae. The distribution of this animal, as you can see on the map, it's on the south coast of Papua New Guinea, Indonesia, Papua, and northern Australia, so this tropical region here. This animal, the status of this animal at UECN, it's near threatened now. And the habitat of this freshwater turtle, it's lowland waterways, billabongs, and seasonal wetlands on the coastal floodplain, as the first speaker has covered a little bit. So in the dry season, this turtle buries itself on the mud and estivates for a few months. It's how it survives to the dry season here in Australia. And when is the good time to find the turtle. So the diet of this turtle, it's carnivorous. It swifts from crustaceans to fish, tadpoles, frogs, and aquatic insects and invertebrates in general, aquatic invertebrates. And that's an interesting point for this species to investigate heavy metals, because as a carnivorous, the long-necked turtle, it's on the top of the food chain. The other one that could be higher would be the crocodile, or even the humans. So Aboriginal people love to eat this species of turtle. So because of them, and because the harvesting and the preparation of the turtle, it's a vital element for Aboriginal culture, we went to the field, me and my supervisor, who is Carla Isenberg, and we were interviewing these traditional owners. As you can see in the first picture here, we have an elder woman. This is a businesswoman here in Australia. She's probing the mud with a stick. That's the traditional way to find the turtle. And she heard a knock. It's because she found the shell, and then she grabs. And you can see the turtle. Here's a juvenile on her hands. And then they cook it. They cook it, the whole turtle. They eat the whole turtle, but not the shell. So in the past 6 years, the traditional owners from the Finniss River, they have expressed concerns about this turtle. So according to them, we have done some interviews. Since 2008, these turtles are tasting different. They are skinnier. The yellow, the fat is yellow, not green. They used to call the turtle as the bush avocado. So it's a very beloved turtle. They have noticed some differences on the health of the turtle. So we went to the field and we got the threat assessment, but that's too broad. So I came here to talk only about one hypothesis, that's metal contamination coming from a mining site. The mining site, it's Rum Jungle, uranium copper mine that starts on the 50s and it was running until the 70s. So the Rum Jungle mine it works here in Australia to supply nuclear weapons to US and British government. It was the major industrial development in the northern Australia at the time, and it got very famous for its high pollution in the river, where all the biota has disappeared on 70s. So on the 80s, they started the first rehabilitation, and they say they got 70% of the environment rehab. Not only the destruction of the environment, the mining also was built actually in a sacred site, woman's sacred site for Aboriginal people. So we still now, they are trying to rehab. This year, they start a new project where they are investing more than 200 million Australian dollars and to try to clean the water actually and the rock waste as well. So the methodology, here on the map, you can see where is the Finniss River floodplain, where we have been working. The Rum Jungle, it's 60 kilometers from the floodplain and 100 kilometers from Darwin. This picture here shows the open cut mine on 50s and how it was. The methodology was really simply, we just joined the traditional people to go for harvesting their turtles. That was in August of 2018. So pretty much like this time of the year, it's good for them to find them. And then we found in two sites, 4 alive turtles in one site where we collect carapace, skin, liver, and fat before they cook and eat. And in the other place, we found dead turtles, 17 dead turtles. And then we collect from the shells, it was only empty shells. And then we could collect carapace and skin only. So these samples, they were analyzed in laboratory here in Darwin for those metals here. And for mercury at ANU with Larissa Schneider. She has made the analysis of mercury for us. And with some analysis, statistic analysis, we could compare the areas and all the tissues as well. So the results, we had 21 turtles, as I said before, and 36 samples. The mean LCL was 172.5 millimeters. And among dead turtles, 41 were juveniles and 59 were adults. And these results corroborates with Kenneth 2014, who has done the population structure study with the same species. And here we have the raw results for all the metals, all the tissues collected in both sides. That's complicated to understand, but that's the mean, and then the deviation, maximum and minimum. And this slide is just the results that we got from statistic analysis. So chrome, manganese, and zinc follow the pattern carapace, skin, liver, and fat. Iron and copper follow the pattern liver, carapace, skin, and fat. Cobalt, nickel, cadmium, and lead, skin, liver, fat, and carapace. Mercury were higher in levels, higher levels on the carapace than skin. On site 1, where we had the healthy turtles, I mean the live turtles, with fresh tissues, we had liver, skin, fat, and liver, skin, and fat. And on site 2, analyzing the shells, we found that carapace had higher concentration of heavy metals than skin. The statistics analysis shows some differences among the sites on skin, because it was the only tissue we had in both sides to compare. So chrome, zinc, cadmium, and lead had some significance. On site 2, when we compared the tissues, we had all the metals with some difference, but not iron and nickel. And on site 2, we could compare skin and carapace, and then we found difference on chrome, nickel, and zinc. So what we can conclude for a few points from these results, based on the generally expected levels by the food standards Australia, New Zealand guidelines. However, these guidelines are for fish. We don't have guidelines for turtle meat. it does not appear to pose a risk for humans, or I mean the indigenous people eating these turtles, not for the amount of turtle they consume. The harvesting of the turtle here is very sustainable. So it's just a period of the year they can eat it. The mercury was higher in the carapace, which is not a consumed tissue. So it was higher than the skin. Of course, the skin has keratin as well. So we can understand by this order in the literature that the keratin from the carapace can accumulate more mercury because of the scutes on these keratins. The keratin is a metal binding protein, easily accumulates mercury. Just checking my time. So that's lead and cadmium were higher on site 1, where we found these 4 alive turtles. And this site is further from the Rum Jungle mine site. So how could we explain that? It's because of maybe metal mobilization through the floodplain, or it could be a result from the rehabilitation from the Rum Jungle East branch, which is closer to site 2 on 80s. Metal concentrations pattern liver skin fat are similar to other freshwater turtle species and some sea turtles as well. Metal concentrations are usually higher in liver tissues because this organ is responsible for detoxification, and that was actually very expected. And of course, we had 6 months to develop this project, so we found some interesting results here, and we need further analysis, including collecting turtles, and that's what we are in the process now, to get funding to keep doing this work and collect more and see if it is safe and how safe and how much people can eat. The turtle looks okay, and would be interesting as well to make a physiological analysis of the turtle to see if it's everything okay with them, with the population. So these are my partners. I would like to thank you, all of them, and all of you guys as well to be watching. And that's me. Just here I have the chat, some questions, right? Question 1. Historically, have there been any other kind of turtles that have been contaminated with metal and people have seen similar conditions? Not really. I didn't find anything about skinnier and yellow fat. They always say the fat it's bubbly, so totally and yellowish, not more for white, not so yellow and green like before. So that's something really interesting we have to investigate. And that comes from the communities, not only one community, some faraway people that lives and eats and really get some protein, not the main source of protein, but it's a good source of protein, and also iron. So I invite you guys all to watch. In 5 weeks, Carla Isenberg talking about the iron we found on this species, the same turtle. And that's really interesting point that she will be talking about. I won't say anything, it's just a merchandise for her. Question 2, what part of the carapace was specifically tested? Was it bone, marrow within the plastron bridge, or scutes? The scutes only. So we can't collect bone to do this analysis because it gets different. They are different tissues. So we have been collecting only the surface of the clean carapace, taking only that keratin, like the same material would be the hair or nail. One more, creatures unseen. Did they compare heavy metals in water samples of the habitat or prey species versus what was in the turtle tissues to access bioaccumulation? Not really. This is not a bioaccumulation project. That's the next step. That's what we want to do. We want to collect the water. We want to collect also the sediment along the river. Since the Finnish River, would be interesting to go a little bit in the bottom of this river before the rum jungle. It's a small place there because they start the mining right in the beginning of this river. So we want to compare the samples from sediment to the turtle's tissues and also all the food chain for this turtle. And that was me, guys. Just wait a little bit more.
And that is week 2 of oral presentations. Thank you to everyone for joining us. And we want to remind you to join us next week at the same time, 11 a.m. Eastern Daylight Time. And be sure to go to our website and check out the poster presentations for session 1, which will be open until September 2nd. You can comment and interact with the presenters there. And for anyone who tuned in to see Patrick Maldawan, he is out in the field and attempted to join us, but was unable to. And we are going to work to put him on the tail end of another session so we can see his extremely interesting presentation. Thank you very much, and we will see you next week. Bye, guys.